Today's lecture is the first in a series of lectures continuing on with the topic of transcription, but looking at transcription specifically in eukaryotes. This particular figure on the first slide is one that we've looked at previously, and I just want to review some of the concepts here as we transition in talking about transcription in eukaryotes. The first thing that I want to mention or review for you is the definition of a gene. A gene is defined as a unit of inheritance that occupies a specific chromosomal location. Typically, it is a DNA sequence that encodes or codes for a single polypeptide. However, the true definition, or an expansion of the definition, is that a gene is a DNA sequence that produces a functional RNA molecule. So in the process of transcription, we are copying genes to produce RNA molecules, and in most cases, or in what we've typically thought of as a gene, is what we would refer to as a protein coding gene. However, there are a number of genes that are transcribed, but the result is not a message that is used as instructions to synthesize a protein. These are referred to as non-protein coding genes. So if you direct yourselves to the figure, you can see that we have the central dogma on the right-hand side, which is the way we typically interpret the flow of information. DNA being the storage molecule that contains all of the instructions necessary for life in the cell in question. That DNA as the storage molecule contains genes. Genes are then transcribed or copied on a gene-by-gene basis to produce RNA molecules. At the top of the slide, you can see the traditional categories of RNA that we think of as products of transcription. The first one is messenger RNA, or mRNA, and this is the product of transcribing a protein-coding gene. However, there are two other categories that you should have heard of previously, known as ribosomal RNA and transfer RNA. These are both RNA products, the result of transcription of genes that are non-protein coding genes. The result is, in fact, a functional RNA molecule. This first category, highlighted as point number 1 on this slide, suggests that RNAs are transcribed, or these categories of RNAs are transcribed in both prokaryotes and eukaryotes, and that is true. In point number 2, you can see that there are some types of RNAs that are produced only in eukaryotic cells, and those are the things that are highlighted in the center of the slide. These are additional functional RNA molecules that are the product of transcription, specifically transcription of non-protein coding genes that results in functional RNA molecules. And you can see a list of those RNA molecules here, again, on the center of the slide. So for the point of the next few lectures, we will be discussing transcription in eukaryotes and how that differs from the prokaryotic model that we discussed previously. The first thing that we want to discuss are the RNA polymerases in eukaryotes. And yes, I say polymerases plural because there are generally 3 broad categories of RNA polymerase in eukaryotic cells. They are known as RNA polymerase 1, which is involved typically in transcribing or producing ribosomal RNA, so transcribing ribosomal RNA genes. RNA polymerase 2, which typically transcribes protein-coding genes and produces messenger RNA. And then RNA polymerase 3, which typically transcribes, again, non-protein coding genes. The products are small RNA molecules such as tRNA, another small ribosomal RNA, and some of the additional RNAs listed on the previous slide in the center of that slide that we find, again, specifically in eukaryotic transcription. But what I'd like to deviate for just a minute here is talk a little bit about our understanding of the main polymerase that we will focus on, which is RNA polymerase 2. So for the remainder of the lectures dealing with transcription in eukaryotes, we will mostly focus on protein-coding genes, the product of those protein-coding genes being mRNA or messenger RNA. But again, I want to emphasize that there are non-protein coding genes. There are different polymerases involved in transcribing those genes, as well as those products are functional RNA molecules that themselves are not instructions for protein synthesis. But again, we will focus mostly on those protein coding genes. What you see in this particular slide is a common technique that is utilized in order to understand protein-protein interactions. In this particular case, this technique, which we're going to discuss in just a minute, was a technique utilized to understand RNA polymerase II and all of the components associated with RNA polymerase II. In a typical tagging procedure known as epitope tagging, and the epitope is a portion of a molecule that is recognized by the immune system. This method or technique is a modification of our understanding of the immune system, manipulating it in order for us to gain knowledge about other proteins. So the first step in this protocol is to genetically tag or mark a protein in question. So in this case, we want to study what proteins interact with RNA polymerase II in eukaryotes. And so in this specific case, the RNA polymerase II was genetically modified or tagged, and that's what you see in the top portion of this figure. That smaller, bluer blip is meant to be the RNA polymerase II, and then the little red additional component is an epitope that is a tag that allows us to easily grab or identify the RNA polymerase II. The next step is to allow for assembly of proteins that could in fact interact with RNA polymerase II to do exactly that. So what you're seeing is several proteins that interact with one another, but also interact with the RNA polymerase II, congregate or aggregate around the RNA polymerase II. The next step is to use an antibody that can recognize the tag that we inserted as scientists to RNA polymerase II. And this basically allows us to grab the RNA polymerase II and all of the proteins that have associated with RNA polymerase II. The next step then would be to separate those proteins out and to analyze those proteins individually. One way to separate them out would be a modified type of electrophoresis that is specifically for protein separation. The concept works similarly to agarose gel electrophoresis, like we've discussed for DNA or RNA work. Again, the concept is similar. You have a matrix that you allow your proteins to separate through, and you can then work with individual proteins that have been part of this group. So again, epitope tagging, and also known as immunoprecipitation, is a very common molecular tool to allow researchers to study a protein of interest, but more specifically, study the proteins that are associated with your protein of interest. Okay, so let's focus on RNA polymerase II in eukaryotes. And again, RNA polymerase II transcribes protein-coding genes. The product of transcription of those genes with this particular polymerase is messenger RNA or mRNA. What you're looking at here is the ribbon structure model for this particular enzyme. And you can see that this enzyme looks very similar in structure to the RNA polymerase that we looked at in prokaryotes. However, this particular polymerase is made up of 12 distinct subunits that can be divided into 3 groups. One group of subunits is referred to as the core, and there are 4 different subunits that make up the core. These are all related in structure and function to prokaryotic core subunits. So these components are very similar to those ones that we talked about in prokaryotic transcription and the prokaryotic RNA polymerase. There is also a category of subunits referred to as common, and there are 5 subunits in that category. Common just means that they are found in all 3 of the RNA polymerases found in eukaryotes. And then lastly, there is what's referred to as the non-essential subunit category, and there are actually 3 proteins in that category. Non-essential typically has an association with some type of regulatory component. When we use the term non-essential, it means that an organism can survive without these particular components, hence being non-essential. Something that would be considered or tagged essential would be something that the organism cannot live without. This is just a more cartoonized version of the RNA polymerase II molecule. And what we can do is talk about some structural features or characteristics. This particular molecule, as I mentioned in the previous slide, looks a lot like in shape overall as the RNA polymerase that we find in prokaryotes. It has this claw-like structure associated with it. And with this particular example, that comes from X-ray crystallography of the structure, we know that there's a very deep cleft that occurs, sort of like in the palm of your hand, if we're going back to this claw-like structure. We know that magnesium does in fact sit in the catalytic center and is required for the function of this enzyme. We know that the presence of nucleic acids are found in what's referred to as the clamp region. And that's what you see more at the top of the structure overall. We also know that this does close down on DNA during the transcription process, and sort of a closed clamp or a closed claw over the DNA allows for processive transcription, meaning that the polymerase won't fall off and continues to transcribe the entire gene. Okay, so since we will be focusing on RNA polymerase II and the genes that RNA polymerase II transcribes, we're going to focus on the promoters associated with those genes. Now, typically, this is referred to as a class II promoter, and that's because it's RNA polymerase II that associates with this particular type of promoter. When we discuss this particular type of promoter, we have what we refer to as the core. And the core has 4 distinct components associated with it, which we're going to discuss here in just a minute. And with a typical promoter, you will find at least 3 of the 4, if not all 4. So the first sequence component of a typical promoter is known as the Tata box, which is right in the center of the slide. The Tata box sits at the -25 position, and this numbering is exactly what we talked about with prokaryotic cells or prokaryotic genes, in that the +1 site is the very first nucleotide transcribed, and you can see that arrow moving in towards the right of the slide indicating going into the gene. So the Tata box is indicated in the red and sits at the -25 position from the transcription start site. The Tata box plays a very important role in alignment to allow for transcription to occur at the transcription start site. So it is functionally very similar to the -10 box that we saw in prokaryotes. There are, however, some genes that are referred to as being Tata-less, meaning they do not have a Tata box. These are going to be things that are typically housekeeping genes. They're constitutively active, meaning they're turned on all the time. And there are some that are also developmentally regulated genes. However, if a promoter associated with a gene does not have a Tata box, it typically does have at least what's known as the DPE, shown in the blue, known as the downstream promoter element, and I will get to that in just a second. But going back to the Tata box is a very common core promoter component, And specifically, this is where we have the assembly of protein factors that are referred to as generalized transcription factors. So what we're going to see in eukaryotes is that there's a much more complex process of transcription initiation that occurs, which then allows for the regulation of transcription to be more fine-tuned than what we find in a typical prokaryotic cell or gene. So this introduces a set of protein factors, which we're going to get to in the next slide, that are known as generalized transcription factors, and their job is to help with the transcription process. Okay, so since I mentioned the DPE, or the downstream promoter element, we'll talk about that just a little bit. That's indicated in the blue, and it's all the way over to the right on this particular slide. It's typically found about 30 bases downstream of the transcription start site, and it also does play a role in binding generalized transcription factors. So as I mentioned previously, if you have a promoter that is a tataless promoter, there will almost always be a downstream promoter element because, again, these two allow for association of these generalized transcription factors, which are necessary for transcription to initiate. In addition, in the green box, you have the initiator sequence, which is abbreviated INR. It is a conserved set of sequences that are around the transcription start site that play a role in optimal transcription. You also have in the purple box the BRE, and BRE stands for transcription factor 2 letter B binding site, and that's how it gets its name, BRE, because it is the B binding site, specifically the generalized transcription factor letter B binding site. This will become more clear in the next slide. Lastly, you have in the yellow upstream elements. These are similar to what we talked about in prokaryotes. They are upstream, and they do play a role in allowing for the regulation of transcription to occur. And we will talk about some of these in a future lecture. Okay, so moving along, this particular slide is meant to be a little more of an overview of the generalized transcription factors, and then we'll look at them in a little more detail in the next slides. So, as I mentioned previously, we have these proteins or enzymes that are referred to as the generalized transcription factors. Their job is to combine with RNA polymerase 2 to initiate transcription. It is a large multi-protein DNA complex, and I want to talk a little bit about the terminology as it becomes important as we continue on with the naming of these proteins. Collectively, they are known, again, as the generalized transcription factors, but individual components have their names associated with them as well. They are typically referred to as transcription factor 4 polymerase 2, or abbreviated TF, T for transcription, F from factor, the Roman numeral 2 because they are specific for RNA polymerase 2, and then a letter designation A, B, C, D, and so on. Now, in this particular slide, you're only seeing those letter designations, A, B, D, F, for example, but the proper name for each one of the proteins that you see here is TF2A, TF2B, TF2F, and so on. Okay, so now we're going to go ahead and talk about the assembly of these generalized transcription factors and what the specific subunits are doing. The very first thing that you see in this slide is a very simple figure of a gene. You can clearly see the TATA box as part of the core promoter, and you see two protein factors associated with the TATA box. You see TF2D, as well as a protein labeled TBP. TF2D is made up of 13 different subunits. Specifically, one of those subunits is known as TBP, which stands for TATA binding protein. So TF2D, multi-subunit complex, with one of those subunits specifically binding to the TATA box. TBP is highly conserved throughout evolution, meaning that this particular protein is very, very important for transcription in eukaryotes. Its job is to distort the double helix. There are several other subunits that are part of TF2D. Collectively, those other subunits are called TAFs, TAFs, and they are TAFs because they are TATA binding protein, the T, associated A, factors F. So TAFs or TAFs are TATA binding protein associated factors. So all of the other components of TF2D collectively are known as TAFs. Interestingly, these have lots of different functions. Some bind to components of the promoter. Some help regulate TBP binding to DNA. But very interestingly, one of these TAFs has histone acetyltransferase activity. And hopefully you can think back to our previous lectures to understand what a histone acetyltransferase does and why would that particular type of enzyme be important during the transcription process in eukaryotes. Next, you see the association of TF2A and TF2B. TF2A interacts with TF2D, like we mentioned in the previous step, and it helps stabilize DNA and TF2D interaction and binding. TF2B binds to TBP and recruits the polymerase, as well as another factor known as TF2F. So lastly, in this progression, you see the association of RNA polymerase 2 with TF2F bound to RNA polymerase 2. TF2F's job is to target the polymerase directly to the promoter. And as you can see, we're slowly assembling a multi-protein complex at the promoter site in order to initiate transcription. Next, you can see the association of two additional generalized transcription factors, TF2E and TF2H. TF2E is thought to recruit TF2H. It also stimulates activity associated with TF2H. So I'm going to talk about TF2H and we'll come back to TF2E. TF2H is a multifunction enzyme. It does have helicase activity, which hopefully you remember that helicases unwind nucleic acids. So this would be important to pull apart the two strands of DNA to allow for transcription to occur. There is also an ATPase associated with TF2H, and an ATPase is an enzyme that can cleave phosphates from ATP, releasing the energy associated with ATP. And then lastly, there is also a kinase function, and a kinase is a class of enzymes that transfers phosphate groups to proteins. And this typically regulates the function or the activity of the protein that the phosphate is being transferred to. This is going to be really important when we talk about RNA polymerase II function. So going back to TF2E, its job is to recruit TF2H, but also stimulate the kinase, helicase, and ATPase activity associated with TF2H. Now, the last step associated with initiating transcription in a typical eukaryotic cell or a typical eukaryotic gene is going to be modification of the RNA polymerase. If you look at this cartoon figure, you can see the RNA polymerase in purple. Again, it has that claw-like structure that we described previously. But it also has what we refer to as a tail. It's a portion that's sort of hanging off of the end of the polymerase. If you look at the previous figures, you will see that that tail does not have phosphate groups associated with it. However, in this figure, the phosphate groups are associated with that tail. This portion or tail is referred to as the CTD, which is short for carboxyl terminal domain of polymerase II. Typically, again, we just refer to it as the tail. And again, as you can see in this particular figure, that tail has multiple phosphate groups associated with it. It is this phosphorylation of the tail that allows for the polymerase to become active and begin the transcription process. So this figure is a different figure that's representing essentially what we just looked at in the previous several slides. What you're noting in this figure is that you have what's known as a pre-initiation complex that has TF2A, B, D, F, and the polymerase as part of the pre-initiation complex. However, at this particular point, the tail or the C-terminal domain of the polymerase is underphosphorylated, more correctly termed hypophosphorylated. TF2H then phosphorylates serines in the C-terminal domain of the tail in the RNA polymerase II. And this phosphorylation event, or multiple phosphorylation events, is essential for the initiation of transcription. In order to pass from initiation of transcription to elongation, that tail has to become hyperphosphorylated. So very simply in this figure, starting from left to right, you can see the tail with no phosphates on it. In the next panel over, you can see that there are some phosphate groups associated with it. And then all the way to the right, you can see several more phosphates associated with it. And it is this transition from a hypophosphorylated to a overphosphorylated state that transitions from initiation to elongation of transcription. During elongation of transcription, there is another factor known as TEFB, transcription elongation factor B, that is a kinase that allows for this hyperphosphorylation to occur. Now, during elongation, the TF2F stays associated with the polymerase. However, the other generalized transcription factors dissociate and the polymerase continues on.